What if we could learn how to make fuels and chemicals sustainably by studying animals that eat grass? It's not as far-fetched as you might think. Cows, goats, zebras, giraffes, you name it. Lots of these large herbivores evolved to graze on woody materials like grasses and forage that we don't consider to be food. And this is an abundant renewable polymer in our world. These animals have the ability to unlock the energy contained in plant cellulose and then convert it to sugar. Large herbivores do this by relying on a collection of microbes within their gut that work together to break down biomass. Unlike us, herbivores are home to plant-degrading bacteria as well as fungi that thrive in the absence of oxygen. In my lab, we isolate the microbes that are inside of these herbivores that are really good at chewing up plant biomass into sugars, which allow released sugars to be fermented into renewable chemicals from biomass, rather than chemicals sourced from petroleum. I first became interested in this problem when I was a postdoc, and I was particularly intrigued by the wealth of enzymes that are hidden in nature, especially from microbes that traditionally are thought to not be cultivatable. My team goes about doing this research in a pretty non-traditional way. What we do is we go straight to the source, and we actually isolate the biomass-degrading microbes from poop. So we go to the Santa Barbara Zoo and we coax out the biomass-degrading microbes using really old-school lab techniques to enrich the microbes that degrade cellulose while suppressing growth of other organisms that we don't want, like bacteria. My group at UCSB is unique in that we combine these old-school microbial enrichment approaches with new school tools like genome sequencing and RNA sequencing to understand how these unique microbes work. So far, we've identified dozens of new microbes that are sourced from these herbivores that have never been discovered before. And these are valuable gold mines for finding enzymes that we could use in the bioprocessing industry. In addition to enzyme discovery, current efforts in my group work to decipher how gut microbes defend themselves from one another. This offers an exciting new route to uncover a class of compounds called natural products, which could serve as new antibiotics. We seek to learn these rules from nature by studying minimal systems, which allow us to design synthetic partnerships between different microbes that would not normally see each other in nature. This opens the way for entirely new routes for bioprocessing, as difficult breakdown and production tasks could be divided over different microbial players in culture. Overall, we aim to streamline the conversion of plant waste into renewable fuels and chemicals.